The blue budgerigar mutation is one of approximately 30 mutations affecting the color of budgerigars. It is part of the genetic constitution of the following recognized varieties: Skyblue, Cobalt, Mauve and Violet. Appearance: the blue mutation changes the color of the body feathers, which are light green in the wild type, to sky blue, and the color of mask and other parts, which are yellow in the wild type, to white. In the domesticated bird, this mutation changes the light green variety into the sky blue variety, the dark green into the cobalt, and the olive into the mauve. The green coloration of the wild budgerigar is due to the combined effect of a yellow pigment and an interference effect similar to that which gives color to petrol on water, which in the budgerigar produces a blue coloration. Yellow pigment is present in the outer layer, cortex, of the cells forming the barbs of all feathers of the wild budgerigar, with the exception of the cheek patches, although it is very weak in the outermost flight feathers. The distribution of the yellow pigment is clearly shown in the lutino. The blue mutation totally inhibits the production of this yellow pigment, and as far as is known, it has no other effect. The yellow pigment in young budgerigars is paler than in adults, which makes green budgerigars in nest feather appear duller and lutinos appear paler. A brighter and stronger yellow coloration appears after the first molt. The chemical nature of the yellow pigment in budgerigars and other Psittacidae is unknown, and in the absence of a chemical name, George A. Smith coined the term cytokines to cover the yellow, orange and red pigments found in parrots and parrot-like birds. Cytokines impart a far more constant intensity of color to feathers than do the more commonly found carotenoid pigments such as xanthophyll. The yellow pigment found in the canary. At each molt, the canary extracts xanthophyll directly from its food, and the depth of coloration of the growing feathers is determined by the concentration of xanthophyll in its diet. Budgerigars cannot be color-fed in this way, because they do not use xanthophyll as a pigment. The blue mutation provides a widely accepted division of domesticated budgerigars into two color classes: the green series and the blue series. Birds of the green series exhibit yellow pigmentation, while birds of the blue series lack yellow pigmentation. These names can be misleading, since some birds belonging to the blue series, such as albinos, are not blue. Similarly, lutinos belong to the green series, yet are not green. In combination with the dark budgerigar mutation, the body feathers become deeper shades of blue. A blue budgerigar with a single dark factor is called a cobalt, and one with two dark factors a mauve. The World Budgerigar Organization has established precise standards for budgerigar body colors, using the Pantone codes, as shown to the right. Historical notes: the blue mutation made its first recorded appearance in 1878 in the aviaries of M. Limbosch of UCCLE, a suburb of Brussels, but this strain died out, it is believed. In 1881, blues appeared independently in the Netherlands between 1881 and 1885, and a Mr. Pauwels of Everburg, near Brussels, reintroduced them to Belgium from this Dutch strain. The first blues to be seen in England were some exhibited by Messrs. Milsom and Pauwels at the Horticultural Hall in 1910 and the Crystal Palace in 1911. Mr. D. Astley owned blues in 1911, and it is recorded that C. Pelham Sutton of Putney bred a blue in 1912. Blues remained quite rare until the 1930s, fetching up to £100 per pair in Japan around 1928. About the cost of a car at the time. Genetics: the blue mutation is recessive to its wild-type allele, so a bird possessing a single blue allele, the heterozygote, is identical in appearance to the wild-type light green. That is, the presence of a single wild-type allele is sufficient to permit the full production of the yellow citizen pigment. Among the budgerigar fancy, such a bird is said to be a light green split blue, usually written in light green, blue, in a bird which has two blue alleles. The homozygote. The lack of the wild-type allele means the yellow pigment can no longer be produced, and so the body color is blue. The Skyblue. The locus of the blue gene is situated on one of the autosomal chromosomes. The yellow-faced blue eye mutation. The yellow-faced 2 mutation form an autosomal codominant series of alleles with the blue mutation. The loci of the dark budgerigar mutation and the blue allelic series are situated on the same autosome, so the dark mutation is linked to the blue allelic series. See genetic linkage. The crossover value, COV, or recombination frequency between the dark and blue loci is commonly stated to be about 14%, but some experiments have found much smaller values. See genetics in the dark budgerigar mutation. Notes. Taylor, T.G. Warner, C. 1986. Genetics for Budgerigar Breeders. 2nd ed. The Budgerigar Society. Watmer, W. 1951. The Cult of the Budgerigar. 3rd ed. Caged Birds. World Budgerigar Organization. WBO. WBO Color Guide.